Now, clinical cytogeneticists use FISH techniques and other techniques to identify 4 different types of issues that people can face with their chromosomes: deletions of DNA, duplications of DNA, inversions between chromosomes, and translocations between or within chromosomes. So we'll look at one example of each. Here's what a deletion might look like. So far, this looks like a pretty typical metaphase chromosome spread. The chromosomes are shown in white, and there's one FISH probe that has hybridized to 2 spots in the genome. And that's standard for a diploid organism, which this is, like humans. There's 2 copies of every chromosome. So we expect to see 2 chromosomes exhibiting signals for a unique FISH probe like this one. But you'll notice that if I diagram the chromosomes like this, on one of the chromosomes, the one in the upper half of the figure, the FISH signal, which I'm going to denote as a black asterisk, is at the end of the chromosome. On the other chromosome, where there's a FISH signal, it's in the middle. One of the things that this might mean is that there used to be some DNA sequence here on this first chromosome that has recently been deleted, leaving that FISH signal apparently on the end of the chromosome, where on the homologous chromosome it's not on the end, it's in the middle. So the change in location on a chromosome of a FISH probe can be indicative of the presence of a deletion of part of the chromosome. And as I've mentioned, it is possible to have multiple FISH probes that have 2 different sequences. So we could have sequence 2 with a red fluorescent molecule and sequence 1 with a blue fluorescence molecule. These sorts of experiments, looking for duplications and inversions, can really benefit from using multiple colors. So in this example, what these scientists have done is they've got 2 different unique FISH probes. Here they're green and red. And they're looking at a chromosome 15 from one individual and from a second individual. And you'll notice that in individual 1, there's 1 red signal, meaning there's 1 copy of DNA sequence C251-3, and there's 1 pair of green signals, indicating that there's 1 copy of T216-1. But in this other individual, there are 2 sets of red and green signals, which suggests a duplication. There's the original version, and there's been a copy. Detecting inversions also benefits a lot from having multiple colors of FISH probes. In this case, I'm going to diagram the 2 chromosomes that exhibit FISH signals. So this chromosome in the lower half of the panel is the one that I'm going to designate chromosome A. So I'm going to call that chromosome A, and I'm going to call this one chromosome B. And we can already tell that they're a little bit different, because despite having the 2 FISH signals, which suggests that these are homologous chromosomes, they have different shapes. Chromosome A looks submetacentric. Chromosome B looks metacentric. In this case, the white dots that I've drawn on this panel represent the location of the centromere. Now in this experiment, we have 3 different probes, so I'll call them probe 1, probe 2, and probe 3. And because we only have 2 colors but 3 probes, 2 of the probes necessarily are going to be the same color. So we're going to say probes 1 and 2 are green, and probe 3 is purple. If we look at the location of these probes on chromosome A, it looks like the purple signals are above the centromere, and then there are 2 pairs of green signals, one near the centromere and one about halfway down the long arm. If we look at the location of signals on chromosome B, it looks like there's a fish signal just above the centromere. There's another pair of green signals down near the telomere, and in between the green signals, there's a purple signal. The bottom line is the difference in the order of signals, purple, green, green on chromosome A, but green, purple, green on chromosome B, means that although these are homologous chromosomes, the physical structures of the chromosomes aren't identical. There's been an inversion in this case. And what this likely means in this instance is that what used to be chromosome A broke here, where I'm showing this red line, and flipped upside down, and then reconnected to produce a chromosome that looks like chromosome B. In sum, if you have 3 probes and their order appears to change on multiple chromosomes, that can be indicative of inversions. Last, we can look at how fish experiments help us identify translocations. In this case, instead of showing an entire metaphase chromosome spread, I'm only showing the 3 chromosomes that hybridized to fish probes. In this case, we have one chromosome with a green signal near a telomere, another chromosome that has a green signal near a telomere, and then the chromosome that has purple and purple. So when we look at this metaphase chromosome spread, there's something strange going on. Normally we see pairs of chromosomes that have the same fish signal hybridization patterns. But in this case, we still do have diploidy, or 2 copies of every chromosome. You'll see we've got 2 chromosomes that hybridize to the green probe, and we also have 2 chromosomes that hybridize to the purple probe. The strange thing is that one chromosome demonstrates hybridization to both the green and the purple probes. So what's the interpretation of this? This suggests the presence of a translocation, that is, the physical fusion, say, of the telomeres of this chromosome to the telomeres of that chromosome. And if you add those 2 chromosomes together, like that, you get something that looks like the 3rd chromosome. So the way this is strange, again, to summarize, is that we have a diploid organism, but it has 3 chromosomes that hybridize to the probe, not an even number of chromosomes. In a diploid individual, every chromosome should be present in 2 copies. So you should always see 2 chromosomes hybridize to a fish probe. In this case, though, there's an odd number, and that suggests the potential for a translocation to have occurred.